Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to this episode of the Genetics Podcast. I'm really excited to be here today with Dr. Arabella Bouzigues, who is the coordinator of the Genetic Frontotemporal Dementia Initiative, also known as GENFI, and she's a postdoctoral researcher. GENFI, if you're not familiar with it, is an international consortium that follows families with genetic forms of FTD, and they're doing some really interesting work combining genetics, neuroimaging, other biomarkers. I've been wanting to dive into this for a while, actually. So, Arabella, I really appreciate you taking time and looking forward to the conversation.
Hi, thank you very much for having me.
I want to start with your explanation of GENFI, actually. It's, as I mentioned, it's a major international consortium. What is the role that you have involved? And maybe you could tell people a little bit about the consortium as a whole.
Yeah, of course. So GENFI is indeed a consortium of international sites who collect data in individuals who are at risk of genetic frontotemporal dementia. So it's actually been running for the last 12 years, and it involves sites who are based in Europe and also Canada. And it started small and grew over the years, as a lot of things do. And we now have nearly 50 sites who are actively collecting data. The principle is quite simple. Everyone all the sites are actually independent. They have their own funding. However, they all contribute the data in the same way. So they all collect data with a harmonized protocol, and all the data is then collated together and distributed to investigators as a combined data set. And so that is kind of how the consortium has been built and how it continues to evolve. And it is very successful because indeed we do have a lot of data now collected across all of those sites and all of those years.
Can you give us a sense of the scale that you are today in terms of the number of participants across the sites, and also the scale in terms of data types? You have a matrix here, right, of lots of people, but also lots of different data types you're thinking about.
Absolutely. So today we have reached nearly 2,000 individuals. Wow. Individuals are seen yearly. So the principle is that they come in every year. Those people are distributed and can be divided into subgroups. So we have people, kind of a minority of people who are patients. So individuals who we call, symptomatic patients. So they have expressed symptoms of frontotemporal dementia. But we also have their family members who are genetically related to them. So their siblings, their children, and so on. And those people are what we would call maybe at risk. So some of those individuals have a genetic mutation, which, and we can go into more detail on this, will go on to lead to frontotemporal dementia. We call these individuals generally pre-symptomatic cases. And then we have the cases who are not carrying the genetic mutation. So they are the lucky ones in the gamble of genetics, let's say. And they indeed are what we will consider the control cases. So in a way, they constitute the perfect control group for our pre-symptomatic, but also patient groups, because they are family relatives. They have the same background other than this mutation. So that is kind of how we would divide our participants. And as I said, we have nearly 2,000 individuals now who have been involved in GENFI. When it comes to the data types, we do indeed have a large panel of data types. So we collect all the usual and classical clinical measures, whether they be by, questionnaires given to the participants, questionnaires given to their caregiver or person who might see them the most often. We also have questionnaires which are administered by a clinician. We have a range of cognitive testing. So again, those are quite diverse, some questionnaires, some actual tests, which can be kind of gamified or not. Some of them are even digital also. We have a range of, all participants do an MRI scanner. So we have a range of sequences that come from that, an array of different types of sequences which tap into different things. So both anatomical, but also functional scanning. And then we also collect biofluid data, so blood and also cerebrospinal fluid for those who are keen and willing to do a lumbar puncture. And of course, genetics, which we get obviously from blood. We can do all sorts of genetic testing as well, which obviously is part of the study, as we do have to test if they're carrying the genetic mutation, but also other genetic elements can be assessed. And we also now have more and more digital tests. So things like eye tracking, smartphone kind of apps that detect different things. We also record voice. So we record their voice during a few short tasks, things like that, which we're trying to develop a little bit more at the moment.
Wow, amazing. There's a lot of areas I want to dive into with all of those, actually. I want to start, though, for people who aren't as familiar with FTD, if you could just talk a little bit about what does early FTD look like when someone who's pre-symptomatic, maybe starting to get their first symptoms, and then if you could just at a high level talk us through the progression of the disease and what it looks like over time, so everybody can get an understanding. Yeah,
absolutely. I think it's really important to understand FTD is very different to Alzheimer's disease. It's, a neurodegenerative disease, so we have that in common. The principles of the pathology are neurodegenerative, where a protein accumulates in the brain for decades before the symptoms then start to show. However, the symptoms are very different to what we know in Alzheimer's disease, for example. So frontotemporal dementia really affects the frontal and temporal lobes, hence the name. And the symptoms that are associated with this disease is indeed, there are kind of different variants. So the most common variant is called behavioral variant frontotemporal dementia, or BV-FTD. And behavioral variant affects, as the name suggests, the person's behavior, their personality. And this is probably why your question is very interesting, is because this variant is obviously something, you could think that personality is quite fluid and that people might change, with time, with age, with life experiences. So indeed, someone isn't necessarily set in a way for the whole of their life. So when there are early symptoms, it is sometimes quite difficult to pick them up because they might just come across as just different, but that might be due to something else. And also it's difficult for you to necessarily assume or think that a neurological disease would affect someone's personality. It feels like that wouldn't be possible, but indeed in behavioral variant, we find that people's behavior is drastically affected. And so the main symptoms are they develop kind of an apathetic behavior. So apathy is when you struggle to initiate and motivate behaviors and actions. Yep. So apathy can sometimes look a bit like depression. And that's often what people might think in the first instance. So if someone is at the pre-symptomatic stage, they might start to be converting to a symptomatic stage. They might actually look a little bit depressed. There's also another type of symptom which is quite common and which is part of the kind of criteria is disinhibition. So they become sometimes quite socially disinhibited. So they might start to say non-appropriate things in front of people. They might not have the right kind of responses to events. They develop also this kind of lack of empathy, which is also quite weird. And again, feels like something that wouldn't be affected by just a pathology in the brain, whereby they might lack empathy, notably with their partner. And that's when it often comes up in the relationship and starts to cause kind of relationship issues or work issues, especially the disinhibition can cause kind of problems at work. So all of that is very subtle. and it starts progressively. And so it's true that you wouldn't necessarily immediately think there's a neurological disorder. So that's the most common variant. And then there's all the what we call primary progressive aphasia, PPA, and those primary progressive aphasia, as the name suggests, aphasia means language difficulties. And we have a couple of variants within that category which affect your language. So people might struggle to name things, and they might struggle to express things. So things might be, the words are difficult to come out. And all of those kind of symptoms are part of this kind of primary progressive aphasia syndromes, which are, again, you wouldn't necessarily think neurological disease immediately. You might not notice the subtle, it might come across as forgetfulness. They might not be able to remember the name of something. And then progressively, as it becomes kind of more and more severe, then that becomes more clear that there's a problem that needs medical attention, and a diagnosis might be established later on.
And when we talk about variants here, my understanding is these are variants in a clinical sense, but there is also some relationship to certain genetic variants it can result in either branch of the tree, right? But maybe you could talk a little bit about what we know about that.
Yes. I mean, the picture of frontotemporal dementia is quite complex in terms of, so as you said, so we've mentioned from the get-go, there's this kind of genetic side of things, which only affects 1/3 of FTD cases. So we say about 1/3 of cases are caused by a genetic mutation. And indeed, most of these genetic mutations, well, all of them can cause behavioral variant FTD. And most of them can also cause a primary progressive aphasia, so a language variant. We also have another set of variants which are kind of more motor related, and which link also to a motor neuron disease, because in 2009, it was found that the same mutation can cause either frontotemporal dementia or motor neuron disease, or a combination of both, which can either start with one and develop into the other, or vice versa. And so that has also enabled us to develop this clinical spectrum where we go from a behavioral variant, a language variant, all the way through to motor variants. And indeed, all of the mutations can kind of cause kind of most of these variants, most of these clinical kind of phenotypes. And then comes also the pathology with the genetic mutation can sometimes obviously dictate which pathology, and then the pathology can sometimes lead to a specific form of clinical presentation. So it is quite complicated. There's no direct relationship at this stage. And that is some of the work that we're doing, is also to try and understand a little bit more, why do some people who have this mutation go on to develop this form clinically, and vice versa. Another genetic mutation might go on to develop another form. So it is a bit complicated still, and we don't have any direct relationships that can enable us to predict things.
Yes. Okay, great. I think this is a perfect segue into there's some very recent work using MRI that we can use as a lens to this, but more generally, FTD can have a very long pre-symptomatic and prodromal phase, right, where these symptoms start to show up, and in some cases it can be a little bit of a diagnostic odyssey, and it makes sense in retrospect for families or individuals, but it can take a long time to get there. And so one of the key areas of research is early biomarkers that are a little bit more tractable than some of these clinical symptoms, which could be easily mistaken for something else. So you mentioned MRI, cerebrospinal fluid for neurofilament, blood biomarkers. It'd be great if you could, and maybe dive into some of the work you've done, like the recent MRI cortical microstructure work, to give a sense of what we know today about the early changes that start to happen in the brain that could be a link to earlier detection and then ultimately earlier treatment of this disease. Absolutely.
So neurodegenerative disease, I mean, this is something that's been very pioneered by Alzheimer's disease. We know that pathology accumulates in the brain decades prior to symptoms. And this was something that we call them the Jack et al. Curves. So Jack, there was a few papers by a researcher, Jack, which show these kind of biomarker changes, and there are these curves that show how biomarkers kind of increase, show signs of change in the brain well before symptoms, and there are different indeed types of biomarkers that one could use. Sometimes we think biomarkers just means like a fluid biomarker, but indeed a biomarker is just something that indicates that there is a change in the brain. And so these can indeed be measured in multiple different ways. And this is really very much the work that GENFI has been kind of leading and really emphasizing. So indeed, things like, well, typically fluid biomarkers, yes, they are very relevant, but the issue is that they are sometimes not that specific. So for example, we found a kind of a marker which has been used a lot is neurofilament light chain. I'm definitely not an expert in that field, but it's something that is indeed a marker for neurodegeneration, but can be affected by other things as well. So it's one of those things where you have to make a decision as to whether or not this is something that we think is specific enough that we can use and base a lot of things off this change, but I think in general, the idea is that we want to complement 1, 2, 3, 4 biomarkers with as many as we can to understand the bigger picture and to use different modalities. And indeed, neuroimaging, and I'm actually quite interested in neuroimaging because my whole PhD was on neuroimaging in FTD and more specifically in GENFI as well, in genetic FTD. And indeed, there are various markers that we have found in the brain, most kind of simply the gray matter, which is studied using what we call a T1 weighted sequence on MRI. And we can really measure the volume of, so it's the neurons, right? The gray matter is the neurons. And so you can really look at how much, quantify the density of the neurons and find out, where there are neurons that have kind of, where there are regions where there are less, fewer neurons, and therefore we call that regions of atrophy. And so this is something that is, I mean, what's important to note is obviously MRI markers are necessary for a diagnosis of FTD. Generally, we look for atrophy of the fronto or temporal lobes. And so what we found is that indeed in pre-symptomatic carriers, we already find this reduction of gray matter. And this, these individuals can be 30 or 40 years old. So they are kind of, 5, 10, 15 years before onset of symptoms. And so, we find a reduction of gray matter. Now, this is where, and there are other markers as well in neuroimaging that can show a reduction or an increase or a decrease of something before symptoms. But it is indeed important to note, and this is something that actually I have a paper that just got accepted yesterday, which will be out soon, and which also looked at gray matter using 2 different metrics. So we used what we call cortical thickness, and we used also a volume, gray matter volume, which is also very typically used. And what we find is that, I mean, the pattern is obviously very overlapping in those 2 metrics. We find the same kind of, reduced cortical thickness and reduced volume in the same kinds of regions, but we do find some discrepancies. And what my paper, at least we tried to kind of argue, is that genetic FTD, these people have a genetic mutation and they've had it from before birth.
Yes.
So their brain has been built differently. Obviously, it can be built differently for lots of reasons, but at least in terms of this mutation, which is involved in all sorts of processes within the neurodevelopmental processes, this mutation has been there from the get-go. And there's a kind of discussion as to whether or not these building blocks might be built maybe slightly differently in the first place. And so it's a very fine line, and this is something that I'm quite careful with when I kind of write up results. In pre-symptomatic carriers, I don't talk about atrophy necessarily. Atrophy meaning gray matter like loss, neuronal loss. I don't necessarily talk about atrophy because I often just say they have a reduced gray matter compared to controls. Because in reality, is it neuronal loss? Has there been a process of neurodegeneration? Or have these individuals actually, from the get-go, had smaller frontal lobes, reduced volume in those regions, because maybe that mutation has been at play? And I think it's quite, one has to be careful what is actually a process of neurodegeneration and what is a process that might just be a difference from the start. And actually, the easiest way one could try and solve this problem, and that is something that GENFI is working towards now, where we've already started collecting data, is looking at children. Because obviously the earlier you go, and some studies have looked at kind of individuals who are younger than kind of 30. So we had individuals who are between 18 and 30 and kind of zoomed into those populations to see. You really wouldn't expect neurodegeneration before 30. I mean, that is not just a decade or two before symptoms, that's more. So I think if you do find differences, and we do find differences in those early carriers, early pre-symptomatic carriers, and again, if you find the same thing in children, you might be asked, the question might not be exactly the same, and you might realize that actually the differences are not just signs of neurodegeneration, just signs of change from the get-go. And that's when your biomarker is therefore no longer a relevant biomarker for detecting neurodegeneration, right? It's detecting a mutation, a difference. Right, great. But we can just do a blood test for that. However, we need to refine our neuroimaging measures to make sure that what they're measuring is actually a neurodegenerative process, something that is going to be a sign of neurodegeneration, maybe something that's going to be a sign of the type of symptoms they're going to go on to develop, maybe that's going to be a sign of the age at which they're going to develop the symptoms or the underlying pathology and so on.
Yeah, this is really interesting. And the question I had actually thinking about this opportunity as MRI becomes more available, but also genetic testing to understand this from small children all the way through to adults. How apparent are these? I saw in the paper that you had actually mutation-specific cortical microstructure signatures. Are these things that someone who's skilled with an MRI can detect by eye if you put the GRNs on one side and the C9s, or is there a kind of complicated pattern that you need AI or ML to understand? And maybe you can talk a little bit about how should we think about cortical microstructure and how you measure it and what it looks like?
So indeed, this is actually quite an important question because it touches also on things that we need to kind of progress on in the future. A lot of this work has been done on its kind of group level analyses. So at an individual subject level, it's not necessarily, well, firstly, the atrophy or gray matter difference is not going to be necessarily visible, just looking at the scan, that's for sure. And on top of even at an individual subject level, but then because these are group analyses, it might just be a tendency for the group as a whole, and everyone might not confirm that. So no, and we haven't in these particular analyses that you're mentioning, or the work that I've done, which has also found these kind of early changes. We haven't used machine learning to identify these. Of course, we have done other work using machine learning, but that hasn't been necessary to detect these particular changes. And indeed, I mean, this is something that the first paper by John Rohrer in 2015, replicated also by Dave Cash in 2018, showed was that there was a genetic group pattern specific to each genetic group of grey matter changes. So like you said, progranulin mutation carriers don't have the same profile of changes in the brain or differences in the brain compared to individuals who have an expansion in the C9orf72 or MAPT tau mutation. So those are the kind of three main groups that we have. We do find that there are specific patterns which affect regions differently. The work that previous people have found, and also that I've kind of just had the paper published in the next couple of weeks, found is that indeed the difference is a lot earlier and a lot more widespread in C9orf72 mutation carriers. And these individuals, the effect size remains quite small. So going back to your question, is this something that you would be able to detect? Just looking at the scan, no. And looking at one individual, probably not either, because it is quite a low effect size. So there are some individuals who are possibly driving this more than others, or because of them all showing the similar pattern, then we are seeing an effect, but it might be just tiny, tiny, tiny. So yes, indeed, we do find very early changes. But in individuals, for example, the progranulin mutations, they actually don't show that early changes. At least it's not as widespread in the brain. It seems to be a little bit more focal and a little bit more aligned with symptom onset. So yes, there is a very different both pattern in terms of within the brain, but also in terms of the stage at which we're seeing those differences in each different genetic group. and they aren't, drastic at very, very early stages, but we can find them at the group level.
Yeah, that's interesting. And it seems like, to me at least, you're painting a picture where there is no one biomarker that's perfect here. And the more of these you have, if you have genetics and you have neurofilament light chain and you have MRI and you have some of these interesting, voice or cognitive tests, then you can start to probably separate signal a lot more clearly than any one biomarker alone, hence the enormous effort you all put into this consortium.
Yeah, we always say this is exactly right. We say that we're agnostic to which modality or even measure within that modality might be useful. And I do feel like indeed a combination of different metrics and modalities will be necessary to paint a proper picture, but also depending on what you're trying to say. Like I mentioned, some measures might be neurodevelopmentally driven, and or some others, and so you need to use them for depending on what you need. Some might be more useful for than others. So whether it be entry criteria for going into a trial, whether it be a measure to monitor if the trial is working well. So for that, you need a biomarker that's going to show change quite quickly and that's going to show responsiveness to the treatment. So you want a different biomarker maybe for that than the one that you use for entry into the trial. And again, to assess how useful the effect was in the long term, then maybe you want a kind of very much patient-driven feeling or caregiver, feeling of change, or what we call like a significant for the patient, a change that is significant for the patient, and not just, we've not lost a couple neurons,
right.
Great. Yeah. So
someone needs to look at it and go, wow, right?
Exactly.
Have that wow. Yeah.
Exactly. So I think all of that means that the field of biomarkers is wide, and I think it should be, and it will continue to be until we have, a treatment for everyone. And again, the fact that it's different, and obviously in genetic FTD, we're looking at often gene therapies. So again, the biomarkers and view of what we've found in terms of the pattern of change and also some of the links with the clinical presentation, then obviously we're going to be expecting that the biomarker will be adapted to that genetic mutation group. So I think, yes, a large panel is definitely the way to go about it. And as researchers, we're really exploring, at least in GENFI, we're exploring everything in the hope that we identify lots of different things that can be useful and which we have already done, of course.
Yes. What has been the hardest things behind the scenes to bring all this together? More than 50 sites, probably dozens of countries. What does it take to pull this together? Because there are not... there are the unsung heroes like you, I imagine, and many others who are behind the scenes figuring out how to harmonize data and figure out data transfer cross borders. Maybe you can talk a little bit about that.
Yes, it does involve a lot of different kind of challenges for sure, of different natures. Indeed, you pick on a very valid point, which is the data transfer agreements, which are not the easiest transatlantic or even between countries that are closer. So yes, I think that is a big hurdle, and luckily, so the way that the consortium works is that we do have kind of a managing, kind of coordinating team, which is based at UCL, and which is the team that I'm part of and that I've been kind of coordinating. So I think that has been one way of kind of simplifying things, is you kind of have one core group that kind of manage the others, if that makes sense. And luckily we have, lots of sites, but they all very, we have a great relationship with them all. And so that kind of trust and that situation has worked very well. And I think indeed, the difficulty, I think the difficulty is managing the sheer numbers now, because, originally it grew organically. So it started small, and originally it wasn't, such a huge amount of work, because it was, obviously the setting up was, but then managing the data was more, relatively simple and straightforward. But now we have more and more data, more and more different types of data, and we have indeed a huge number. And so that is a lot, and has meant that we've had to think about how we do this differently and evolve with the data. So, before it used to be an Excel sheet that would be manually kind of managed, as is a lot of clinical data. And then with time, we've now developed, scripts which kind of automate things as much as possible to reduce human error. Again, the way that we kind of input the data, we still have a bit of manual integration of data where people have to manually input, scores on tests or answers on questionnaires. But we've tried to reduce that as much as possible with some things that go kind of automatically get uploaded, things like that. Again, I mentioned earlier that we do have quite a few digital initiatives, which again, automatically kind of link up. But yes, I mean, the main challenge has been kind of technically getting that all in place, yes. Getting the databases and the setups kind of working for everyone. That we always have problems with sites who are struggling to use some database of some form. So it's very much a coordination process, and as I said, led by one kind of small unit. I think the main, and it sounds silly, but it's obviously important, difficulty in GENFI is that we don't have one funder.
Oh, interesting.
We have, there's no one funding body that has funded anything in GENFI. It's been every site has got their own bits of money, often a jigsaw of lots of pieces from here, there, and everywhere. A bit of clinical money here, a bit of fellowship money here, a bit of grant money from there. And together, this has, funded their own local data collection, whether that be their MR physicist locally who coordinates with our UCL team to make sure they've got the right sequences, their neurologist who assesses the patients, et cetera, et cetera. And at UCL, we obviously have all these additional costs of this managing team, which has been small, very small, and junior. I'm the most senior member of that team other than Professor John Rohrer. And, it's small and it's mostly made up of research assistants. And, it's very homemade, let's put it that way. But it also, comes with that a real keenness to do things well. When something's homemade, then there's that nature of things that are done very well and with the best interest possible, So I think that is something that's very important in GENFI. And we have a really great relationship and connection with all the centers and all the sites and all the members. So I think, they trust us to help them when they have a problem. We trust them to be able to solve the problems with our help and collect the right kinds of data. I think it's a nice group, and it has been but it has organically developed. And I think we're thinking of going into a new kind of phase, which will be even more kind of connected with tools that can do as much as possible kind of in an automated fashion, so that we can both reduce the workload and the burden, but also reduce human error and also enable us to collect more data without it being more work for everyone, because we just don't have the funding, to fund help for everyone, technicians for everyone, etc. So yeah.
Yes. My call to action to any philanthropists or funders listening is reach out to Arabella if there's anything you can do here.
Great pleasure.
It is one of the things that's always struck me about working with ALS and FTD communities is one of the things you really have going for you is I think people, something not to take for granted that isn't always the case in other fields, is that these are such devastating diseases. There is so much unmet need that there's a lot of drive to collaborate, and so this is something I think that can come together in a field like this, where people say, even if funding is a little tight, like, let's figure out how to do it, because we have to, right? There's just so much unmet need that you can't not do something.
The field is amazing. This field, I mean, I don't know that many different fields, because obviously for now my career has been kind of mostly related to FTD research, but I've got to say people are so committed, whether that be the patients themselves, involved, invested. They will give anything. They will have a lumbar puncture. For some of them, they will have a lumbar puncture every few months. Wow. If it's helpful. And, you all know how, or don't know, but you can all imagine how uncomfortable and unpleasant it is. They are so dedicated, and the researchers in the same way, so dedicated, so selfless, the time, energy, money that goes into this, so many people involved, but everyone does it with this huge amount of optimism and huge amount of care. Actually, it's it's a really beautiful field, I think. And I think, like you say, it might be specific to this type of disorder, which is so devastating. I think obviously there are lots of others which have that similar nature, but it's true in frontotemporal dementia, and I think in ALS as well. We also collaborate a lot with them, and there is a huge amount of kind of a really great relationship between the different levels involved, the different kind of partners, let's say, and it feels very wholesome and generally very positive, even though to this day we don't have a treatment, but there's a huge amount of positivity, a huge amount of encouragement, which is really, really pleasant. And like you say, the collaboration comes naturally. There's no need to insist, or you don't need to try that hard. Everyone is very, very willing to collaborate. I think it also comes from the fact that it's a rare disease as well, so we have to collaborate if you want to go anywhere, whether that be the patients or the researchers or industry. So I think that is really a lovely side to this work.
Yeah, you perfectly segued, I think, into the last question I want to ask you, which is about treatments and therapeutics. As you mentioned, there really isn't one in the community right now. What are you most excited about? Where do you see the greatest promise going forward for FTD in terms of treatments and therapeutics?
I mean, there's a lot going on. There's a lot of movement. There's a lot of desires to try things and work on things. I mean, one thing that obviously we're very proud of in GENFI is that we're working quite closely with industry to try and feed as much as we can from our experience, from our data. And there are several biomarkers that we developed and validated in GENFI that have now been used in trials, whether that be a clinical measure like what we call the CDR plus NAC FTLD NM score, which is used as a clinical marker of severity. We have a cognitive marker, also the GENFI COG, which is a composite of different cognitive measures, which we developed and is being used in trials, and also volumetric T1 measures. So like we said, the volume of gray matter, and NFL, like I mentioned. So we do have lots of things that we validated in our cohorts, which are being used in these trials. And that feels like there's a bridge of communication between academia and pharma, which is obviously really reassuring to feel like the companies are just trying out these tests or these scores without our kind of experience contributing towards it. So that's great. And I think the thing that I'm most excited about is continuing along those lines with more and more of our biomarkers being obviously validated and tested in our cohort and then going out into the trials. We're also so there's been trials in C9orf72 mutations and in progranulin. There are quite a few trials on the go or kind of in motion. There haven't been any trials in the tau mutation, so there's been quite a lot of discussion about developing a platform trial that would be academically led. Cool. So that's something that I think we're quite excited about, because it's all these initiatives that kind of cross the board from the patient all the way through to the industry and the treatment. And I think we can be really a part of that. We have the patients, we have the individuals who need the treatment, we have the experience, we have the data. And so I think all of that means that we can really be invested. So I think that's really exciting. Obviously, I can't say like if we're going to have obviously a treatment, in the next couple of years or in the next decade, but I do feel like the fact that things are at least going in the right direction, the collaborative efforts are in place and are working relatively well. I think that is all promising. And I think that's very much the direction we're going in. I mean, our lab at UCL, so John Rohra's lab at UCL and generally the GENFI consortium, we're all very much oriented towards how can we improve trials success, essentially. And that's what we're working towards. And we do very little work that is purely what we would call fundamental research. We do very little of that work. We're very much interested in trying to develop biomarkers and trying to help develop things that will be useful for treatments. So I think that's what I'm most excited about, and that's where we're going in the next few years, at least for GENFI and the wider FTD community, I think.
Amazing. I share that excitement. Arabella, thank you so much for your time. This has been an amazing episode.
Thank you very much for your time. And I hope that this will have generated lots of thoughts and impressions with the audience.
I have no doubt about that. Thank you, everyone, as always, for listening, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.